Yeah, thank you so much for the introduction and this opportunity to present here today. It will be hard to follow all these great talks, and as everyone was working on CAR T cells, I still want to stay on the CAR thing, but I'll change a little bit of gear and talk about NK cells. This is a really off-the-shelf product, truly off-the-shelf product, unlike all the CAR T cells that has been approved by FDA till date. These are my disclosures. We all know immunotherapy has been a fifth pillar for cancer therapy, initially because of the discovery of this immune checkpoint blockade from these two great scientists, Dr. Allison from MD Anderson and Dr. Honjo from Koto University. But after that, we all know of the remarkable efficacy that is shown by this CAR T therapy. This cancer immunotherapy has been a great pillar. And no need to discuss all these timelines, but we want to say that this CAR T therapy has revolutionized the field, as we all know. But as I earlier said, all the CAR T product till date that has been approved by FDA are autologous. What does that mean? It's just a brief. When patients is diagnosed, you had to undergo apheresis, manufacturing, and then it takes a couple of weeks, freezing and sent back to the center. So I know a lot of research has been ongoing to minimize this timeline. We'll be hearing talks tomorrow, I guess. But despite that, we need to appreciate that it takes time. And not only time, the product too. I know nowadays the product failure is very low, but even though there are some manufacturing failures with the CAR T therapy. So we and many people, what we are thinking is, what if we can really identify a healthy donor, choose your immune effector cells, whichever is your favorite. NK cells happen to be our favorite, but it doesn't have to be NK cells. You manufacture them, you freeze them in multiple doses, so that what happens when the patients come to the clinic, you can directly treat those patients. So what happens with this is it decreases the cost, of course, and it gives access to the patients, and it increases the speed. And one thing is very key here is about access, because the CAR T therapy that we have been giving now, actually you cannot give in those small community hospitals, right? Just because those hospitals doesn't have the expertise, or they don't have the expertise to manage these patients. So it has to be very centralized hospitals. So, and I don't have to talk about this financial toxicity and clinical toxicity. So what we wanted to do, or what we think about in Katie Rezvani's lab at MD Anderson, is how we can make sure that this patient doesn't have to go through this clinical and financial toxicities. So for that, as I said, our favorite cells is NK cells. It's called natural killer cells. Unlike T cells, which has to have this antigen-dependent killing, natural killer cells, they are born to kill. So they have these inhibitory receptors, activating receptors. They find these like virally infected cells or cancer cells, and they just go and kill them. And then we engineer them on top of it to express CAR, so that they can go and identify these tumors and kill them. And a little bit of like they are CD56 positive, CD3 negative, differentiated in bone marrow, like we all know about this. So we had to thank all those great scientists who actually spend years and years to actually study these natural killer cells. And only after this past 2 decades, people have been trying to use these NK cells as a therapy, and we at MD Anderson are leading along with other groups by using it as a CAR therapy or CAR TCR therapy or some other kinds of therapy. So today I'm going to talk about CAR NK cells, and in our lab we always think about how can we make this therapy better, right? So for the interest of today's talk, I'm going to touch upon two topics. One is how can you identify this suitable antigen? As the previous speaker also said, for a CAR therapy to work, you had to have a suitable antigen. And CD19 happened to be one like fantastic antigen for hematologic cancers, but still there is not a suitable antigen for solid cancers and other type of hematologic cancers, for example, AML, right? And other is optimizing the CAR construct, because even though there are a lot of research going on in NK, like clinical trials, what we just did is we just borrowed this vector from CAR T, right, with either CD28 co-stim domain or 4-1BB, especially CD28 co-stim domain, and just used it in NK cells without even knowing like whether this co-stim actually works in NK cells or not. So, I'll be touching upon that. So the antigen we chose is CD70. CD70 is a ligand for CD27 receptor, and they are a very attractive pan-cancer antigen. And it's because the CD70 is only transiently expressed in activated T cells or B lymphocytes, and they are not expressed in normal tissue. And just a brief introduction, how we are tied with the Miltenyi, as everyone is tied with. The NK source, many people use peripheral blood NK source, iPSC NK source, but we are very strictly using core blood derived NK source, just because MD Anderson has a core blood bank and we have access to those core blood banks. So we take the core blood unit, we isolate NK cells, we expand them in the presence of feeder cells and IL-2, and then we transduce them, and then we make our NK cells. And later I'll show you how we freeze these NK cells. So the first thing, CD70, right? So first thing we check is whether it's expressed in hematologic malignancies or not. So first one was AML. As you can see here by flow cytometry, we can clearly see that CD70 is expressed on AML. When we divide AML into leukemia stem cell like a subset or blast, both subsets happen to express CD70 at high expression. Interestingly, just because we are at MD Anderson, we have the access to these patients who has been treated with CD19 CAR T therapy. So what we did is we checked the CD70 expression on those patients which are prior to CAR T19 therapy, and those patients who relapse this CD19 CAR T therapy. And as you can see here, luckily those patients who relapse CD19 CAR T therapy has higher CD70 expression, suggesting that this target can be an alternative target for these patients who actually relapse this treatment. And these are the 6 patients which are actually matched patients before and post treatment. And as you can see here, a significant number of patients happen to express CD70 post CD19 CAR T failure. Next is solid cancers. We take the TMA from various different organs, and as you can see here, the kidney cancer actually has the highest CD70 expression, and more importantly, the normal tissue has very low expression of CD70. This is H score, which is quantified. Here are some representative images. You can see here in normal kidney, there is very low expression of CD70, but tumor has high expression. Similarly, breast and brain and so on. So that makes that CD70 is a very good target. So next what we did is we designed the CAR construct. But here, unlike this traditional CAR construct where they use this SCFV region from the antibodies, the fact that CD70 has its natural receptor, we chose the sequence of the natural receptor CD27 in order to make our extracellular domain of our CARs. So as you can see here, we check the natural sequence of CD27 and make the extracellular domain. And to make our story very simple, what we did is we make the same transmembrane domain and the intra-signaling domain, which is CD3 zeta, but we went ahead and changed the co-stim domain. As we know that in CAR T field, co-stimulatory domain plays a significant role for the CAR T cell effectiveness, and especially they use either CD28 or 4-1BB. What happens happens is actually CD28 is very specific T cell co-stimulator domain because NK cell doesn't express CD28. And 41BB is a bisystric. That means both NK cells and then T cells express CD41BB. And we also included NK-specific co-stimulatory domains such as DAP10 and DAP12. And we did the preclinical studies to validate whether the transduction works or not. All the CAR constructs with different co-stimulated domain, transduction is similar. We use NT, that is non-transduced NK cells, or NK cells transfused with only IL-15 as a control. And one key information I forgot to tell you is all our CAR construct has IL-15 in the construct, just because IL-15 is needed for NK cells to persist in vivo. And there is no difference in cell proliferation between all these CAR constructs. More importantly, when we culture our CD70 CAR-NK with normal hematopoietic stem cells or progenitor cells or normal T cells, NK cells or monocyte, they don't kill these normal hematopoietic cells, suggesting that there may not be any toxicity related to our CAR product. Next, we need to check the functionality of our CAR product. The first thing we did is we took the Raji cells, which has high CD70 expression, and we co-cultured them with our CAR-NK cells and checked the secretion of TNF-alpha, interferon gamma, and CD107A degranulation marker, which is a surrogate marker for the cytotoxicity of the NK cells. And as you can see here, all of our CAR construct increases TNF-alpha secretion, interferon gamma secretion, and CD107A expression when compared to NT or IL-15 transduced cells, suggesting that these CAR and NK cells are functional. More importantly, when we knock out CD70 on these Raji cells, this functionality is decreased, suggesting that there should be an interaction of the CD27 receptor and antigen in order for our CAR NK cells to be functional. That is a short-term assay, 4-hour assay, so we next moved into a long-term assay. What we did here is just an IncuSight assay where we are monitoring these cells for long-term by like live microscopy. And what we are doing is every 2 to 3 days, we're adding fresh tumor cells to this co-culture to see if these NK cells kill these tumor cells in a long-term assay. And as you can see here, most of the CAR NK cells are better than controls, but the CAR NK cells with CD28 co-stim seems to be doing better in multiple models, which took us by surprise because we thought maybe CD28 co-stim won't work on NK cells because just because NK cell doesn't have CD28 expression. So next we wanted to move it into the in vivo model. So in vivo model, we first used the Raji cells, which is a very aggressive lymphoma model, and as you can see here, indeed the CAR construct with CD28 co-stim seems to be controlling the tumor better and increasing the survival of these mice, suggesting that the CD28 co-stim performing better when we put the CAR construct in NK cells. Next, we knock out CD70, and then we do not see this effect. Suggesting that this interaction between antigen and CAR-NK is needed in order for our NK cells to be functional. So next, we chose the AML model, THP1, which is the AML model, and similarly, we see the same results with our CAR-NK cells. With CD28 co-stim, it seems to be doing better. So for solid cancer model, we use BCX010, which is a breast cancer PDX cell line. This is not a PDX line, it's a PDX cell line derived from a breast cancer PDX. And similarly, in this model also, we saw that the CAR NK cells with CD28 co-stim seems to be doing better in controlling the tumor and in prolonging the survival. More importantly, if you see this figure C, so compared to NT and IL-15, when we check the NK cells in the blood of these mice at day 10, you can see all the CAR construct group with all the CAR constructs seems to be higher. That means these NK cells are proliferating and persisting in the blood. But when we check the blood at day 20, all the groups seems to be going down. That means these NK cells are no longer persisting. But when the group with CD28 co-stim has a similar level of CAR NK cells compared to day 10, suggesting that the CD28 co-stim is helping these NK cells to persist longer in vivo. So as I earlier said, CD28 doesn't seem to be expressed in NK cells. Only when we express CD28 as a co-stim domain in our CAR constructs, this is expressed. And what we thought is, in CAR T field, it's well known that when when CD28 is incorporated in the as a co-stimulator domain in a CAR construct, there is a region called PYAP which brings this LCK kinase, and then it helps in the phosphorylation of the downstream signaling. So we went ahead and checked this hypothesis, because other than CD28, all other kinases that are required for this TCR signaling is also present in NK cells. Indeed, that same thing is happening in our NK cells. As you can see here, without any antigen stimulation, our when we express this CAR construct with CD28 co-stim, the phosphorylation of phospho-CD3 zeta is higher compared to the CAR construct where there is no CD28. But when we stimulate our CAR NK cells with CD27 antigen, this phosphorylation further increases, suggesting that this CD28 co-stim is helping in increasing the phosphorylation of CD3 zeta signaling domain. Next, we deleted the PYAP region on our CD28 region, and we see that this phosphorylation is decreasing. Further, we knock out this LCK kinase in the NK cells and transduce these NK cells with this CAR construct without and with CD28, and only in the CAR construct with CD28 domain. It has high phosphorylation, but when we knock out LCK, this phosphorylation is significantly decreased, which suggests us that this LCK recruitment to the CD28 Indo domain increase the CD3 zeta phosphorylation in order for our CAR NK cells to be functional. So this study is published recently, so you can go back and look at the paper. So this is just a mechanistic study of mechanistic cartoon of what I actually said. When we have a CD28 co-stim, LCK comes and binds here and increases the phosphorylation of downstream CD3 zeta and increases the downstream signaling in order to increase the NK functionality. So now let's move the gear, and with that, we chose this CAR-NK construct with CD28 co-stim, and then went through all the processes that you all know for the IND filing and IND enabling study, and then we started the clinical trial for both hematologic malignancy, which is led by Dr. David Marine, and then the solid cancer trial, which is led by Dr. David Hong at MD Anderson. For today, I'm only going to show you some data on hematologic trial on hematologic malignancies. So as I earlier said, we had this banked core blood, and we have a criteria now to identify what's the optimal core and suboptimal core. So we take this optimal core, and then and if you want to study more about that core, we just recently published that study on Nature Medicine, so you can go back and check. It's the continuation of our CAR CD19 trial. So what we did is we take this, we derive these NK cells, we transduce them, we expand them, and we banked them, and we cryopreserve them and bank them. So from 2 core blood donors, we have so far make a dose which is enough for 250 patients at the earlier dose and at the higher dose. And just keep this in mind: the first product, it has around 60%, 58% transduction efficiency, and the second product has 77% transduction efficiency. So when patients come, they undergo lymphodepleting chemotherapy, and then we thaw our NK product and then infuse them at the bedside. So as FDA, we're very conscious with this receptor-based CAR-NK product. So they want us to start with a flat dose of 8 million cells for a human. So just to give you a perspective, for our animal experiments, we use somewhere from 5 million to 10 million NK cells for 20-gram mouse, and FDA wants us to start with 8 million NK cells. And this is just the NK cells, not the CAR-positive NK cells, and so on. So I'll show you a data from this first few patients, which has received a 8 million flat dose of NK cells. And keep in mind, the transduction efficiency is 58%. So actually, this patient only got like 50%, 4 million CAR-positive NK cells. And as you can see here, this patient has undergone several lines of therapy, including like two transplant therapy, like brentuximab, nivolumab, and CD25 ADC, and so on. So this is the PET scan of this patient. As you can see here, pre-infusion, there are a lot of tumor lesions. This is the day 30 post-infusion, and this is the day 60 post-infusion. And these NK cells are showing remarkable efficacy for these patients. And this patient is a Hodgkin's lymphoma. So far, we are currently at the dose level 4. We have treated like 10 patients, and 8 has shown the early efficacy, and then 7 out of 8 patients are Hodgkin's lymphoma. And when we saw this data on the meeting, people are said, we are no more interested with Hodgkin's lymphoma. People are curious, like there are so many products for Hodgkin's lymphoma. So we get that, And then this is the other patient that I want to show you, which is a patient with a refractory CTCL. And this CTCL patient samples also has high CD70 expression. So as you can see here, this patient has also undergone several lines of therapy, and you can clearly see there is a lesion on the skull, on the lymph, like beside the neck, and all these lesions. And this is the PET scan of this patient at day 30, and this patient has a similar PET scan at day 60. I didn't put it here, but I wanted to show you this picture. That's why. So you see, as you can clearly see, pre-infusion, this patient has a lot of lesion on his skull. There is a big lump at his neck, which is completely gone after our treatments, suggesting that this therapy is working on these patients. So with that, I think I was able to show you that CD70 can be a very good, attractive target for a CAR T or CAR NK therapy. And we developed multiple CD70 CAR NK CAR constructs and found that, luckily, indeed, CD28 co-stimulatory domain CAR also seems to be working on CAR NK field or CAR NK therapy. And for mechanistically, we found that this LCK recruitment to the CD28 endodomain, it increases the CAR-specific CD3 zeta phosphorylation. And we have initiated the phase 1 trial for both hematologic and solid cancer, and hopefully we hope that we'll soon enough we'll present the bigger data on the trial for these patients and some patients from a solid cancer trial. With that, I want to thank my mentor, Katie Rezvani at MD Anderson, and it needs a huge team to do work like this all the way from early development, preclinical studies, doing IND-enabling studies, and we have our manufacturing facility, and we treat those patients like by ourselves. So we do all these 360-degree, like a lab where we start from the beginning and we do all the way up to the end. And then we get this patient sample back, and currently we are doing this immune correlative study to understand what's happening in these patient samples. And then research team, GMP team, all the funding agency, but last but not least, the patients and their family who trust on us and then are willing to take this therapy without knowing whether it's going to work on them or not. So with that, thank you so much for your attention, and I'll take any questions.
So, any question from the audience? One coming from anyone?
Thanks for that elegant presentation. The preconditioning is known to increase IL-15 levels in the blood. Are you able to differentiate between IL-15 due to your conditioning regimen and that's being produced by your transduced NK cells?
Great question. Actually, we are not. So FDA wants us to check the IL-15 label before on the product, right? On a drug product. Fortunately, our drug product doesn't secrete that lot of IL-15, only 150 picogram per ml or so. But when we check the patient's blood, the IL-15 level is not that high, so actually we cannot differentiate. But that's a great question, and I don't know how we can differentiate that. We need to think a little bit about whether it's because of the lymphodepletion chemotherapy or because of our product. But that's a great question. If you have any ideas, we'd love to hear.
So could you comment on the persistence of your cord blood-derived NK cells? Because I don't think I've seen any of that, especially for the clinical study. I understand it's different for the xenograft studies, because it's a different setting clearly, but in your patients, I mean, you have an allogeneic setting as you presented nicely in the beginning. So I'm just wondering, so what is the persistence of your NK cells?
Yeah, thank you. It's $1 million question for NK field, right? Because that's we are all thinking about, like why it's not in the forefront, because it's well known that NK cell half-life is very low. It's usually it's persist for 2 weeks, but because we are giving IL-15, we are able to see the persistence on these patients for up to a year. But of course, we are knowing that that number of NK cells that are persisting is not enough, because these patients are, I'm not talking, CD70 is too early to tell because we are just starting, and we haven't had that milestone studies to read, but we have historic data from CD19 CAR therapy. So we have published that data on Nature Medicine paper, but we are able to, not by flow, you are not able to detect these NK cells by flow, but at least by qPCR, you are able to detect these CAR transcripts on these blood. But yes, that's the thing that keeps us awake at night. Like, how can we make our NK cell persist longer and then show the durability of these responses?
Can I ask a second one?
Yeah, sure.
And my other question was, so can you comment about CD70 loss? I mean, obviously, you know, antigen loss is one of the major reasons for relapse after CD19. It's not the only one, of course, but it's a major one. So can you comment about anything? Have you seen any of that, for instance? I mean, I understand for the clinical part it's too early, but you know, for the mouse models, I mean, obviously most of, I mean, at least in the AML models, I mean, a third of your mice would relapse and succumb. In the Raji models, it's all the mice eventually would relapse and succumb. So have you seen antigen loss or mutations?
That's a great question. Actually, I don't think we checked that in mouse models, and because our product, like as you can see here, we are not completely curing even in mice, right? Because of the dose we are giving. Or Raji, just because they are too aggressive, so we are still seeing this antigen. But we are very interested on what's happening in the patients. So this is something that our immune correlative study will give us an idea whether that's the case or not. But we need to understand that because we need to get patients at the early run, we are also enrolling those patients which has very low CD70 expression on them, right? So we will need some time to really understand about that. But yeah, in vivo models, in mouse models, this tumor still has CD70. Our NK cells is not able to clear all the tumors. Yeah.
Any questions?
Okay, you showed that you froze down 250 doses of these 2 donors. So could you share any stability data or comment on that?
Stability, you mean?
Yeah, so how long would they be okay when they are frozen down, or is it infinite?
That's a great question. Like, that's what FDA wants us to do, is stability test every 6 months. And the first product that we froze was almost a year ago. So we have done 2 stability tests. I don't have the data here, but of course they had to pass all the threshold, like CAR transduction percentage, like a viability and everything. So that product is 1 year, and the other product is just been 6 months, because we made one product and then we made another product a couple of months back. So that one we just did the 6 months stability, and they're still fine. And we don't think there will be a problem, because pretty soon, actually, the product 1, we are almost running out of product 1. So now we are trying to make another product, the 3rd product. And the second product will be out soon. So at least for a year, they were stable, and we passed that kind of thing.
Jürgen?
So far, you have done HLA mismatching to to prevent inhibitory signaling, I guess. So for an allogeneic off-the-shelf therapy, it would of course be important to know whether it's really necessary and to which extent. So any idea on that?
Great question. Thank you so much. That tells me that I forgot to tell one key information in my presentation. That is, we don't see any toxicity on our therapy. So far, we have treated more than 100 patients with CD19 CAR, Affimate, like pre-complexed NK product, and CD70 product, and we have other product, like 7, 8 other clinical trials currently happening. So although we have suicide gene, we have never used a suicide gene for like a single patient. So it's very completely safe, no ICANS, no CRS or anything. So going back to your question, that's a great question. So we thought about this when we started the CD19 trial like 5-6 years ago. And for the few patients, we tried to make like HLA match. And then we went and did HLA mismatch, and then we saw the same kind of effect for the HLA mismatch. So by the mid CD19 CAR NK therapy, we don't even check, like we do check HLA, but we don't care about HLA match or mismatch. So it doesn't affect the patient. But going back now in our meetings, I think we are discussing that maybe we can think about going back to not completely masked, but at least maybe some masked, just in order to make our NK cells persist longer. So there are so many things we are thinking now, how to make NK cells better by some modification or something, and then on top of it, maybe just because earlier we had to do that just because people didn't trust us, they're not enough patient, but now we have a luxury that we get a lot of patients. We might have a luxury, because if you do that, we have to get rid of some patients, right? So that's the thing. But I think for certainly for at least for a couple of years, we will do as we are doing without any HLA matching. And it seems to be working in our hand.